So if, okay, is Jen downstairs? Is Jen doing that or is you're not doing that? Let's see. Okay, if a PTM, what is a PTM? What's the definition of a PTM? So if a cell generates based from a gene, the corresponding mRNA, and then this mRNA gets translated by the ribosomes, the first product, so the amino acid sequence that is generated by the ribosomes, is very often not the final product. It's not the final mature functional protein, but a variety of modifications can happen to this amino acid sequence. And because they're all happening after the ribosome has translated the mRNA, they are called post-translational modifications. And what most people think about when they hear the term PTM, at least I would say what most proteomics people think about when they hear PTMs on proteins, they think about reversible addition of chemical groups onto protein, either on the amino acid side chains or maybe also on the end terminal of the protein. So reversible addition of small chemical groups are, for example, phosphorylations, methylations, acetylations, oxidations, all these things that usually happen on the side chains of specific amino acids. Another type of PTM is the reversible addition of another protein at the side chain of a protein. So a modification with a polypeptide sequence. So examples that fell in this category are, for example, ubiquitinylation or sumylation, which are all important PTMs involved in degradations of proteins, for example. Then we have also reversible additions of other complex molecules, which I mean not proteins, but addition of other biomolecules that we have. For example, lipids, attachment of lipids and maybe even very large lipid structures on proteins, attachments of sugars and very complex sugar structures, attachments of ADP ribose. Then we have also irreversible PTMs, and for example, the irreversible cleavage of the peptide chain itself is also considered a PTM. So proteases can cleave off certain sigma peptides. This is by this definition of PTM. There's also a process called intein splicing, where in the middle of the amino acid sequence, a peptide bond can be cleaved or even fused together. Okay, and then there is also other irreversible modifications, like, for example, deamidation. So things that once they happen to the amino acids are not necessarily reversible. Okay, what I want to say with this whole figure is that PTMs are really super diverse, and they also really lead to proteome diversification. So due to all these PTMs, proteins can do what they're supposed to do, and they really change a protein in many ways. Why are PTMs interesting at all? And again, here I focus on the biological PTMs that occur in nature. So the majority of the proteins that we know are modified, at least at a certain time point during the lifetime of a protein. And there are even many proteins that carry not just one PTM, but they can carry 2, 3, or 10. And what we call these various different versions of the same amino acid sequence with different PTMs attached on different amino acids is what we call proteoforms. And you can imagine, so if you have not just 2, but 3 or 4 possible PTMs, or 3 or 4 or 5 amino acids in the protein, the combinatorial number of theoretical proteoforms that can exist really rises like crazy. So what I want to say with that is that the proteoforms can be really, really complex. And what are the functions of PTMs? What do they do? Well, pretty much everything you can think of. Every functional protein can have has also been associated to certain PTMs. So PTMs influence where proteins localize in the cell. With, for example, lipidation makes them localize in the membrane, and signal peptides make them to be secreted. They modulate activation of proteins. They modulate signal transmission, cell-cell interactions. Informational changes are happening up on PTMs. You can change solubility of proteins, stability, and so on and so forth. So, and unraveling this PTM code on proteins, and maybe even the existence of different proteoforms, is really one of the great challenges that especially also proteomics people face at the moment, and where a lot of research is ongoing. So how can PTMs be analyzed? There are two major ways what people do in the field to investigate, identify, and quantify PTMs. One is the antibody-based method. That means you need an antibody that is specific for a certain PTM type, or maybe even for a specific PTM on a certain protein in a certain position. And then you can do Western blots, Luminex assays, or protein arrays. And the advantage of antibodies is the advantage every antibody assay has. It can be very selective. You can also do it with these arrays in high throughput, but you need to have this specific well-working antibody, which especially for PTMs is something that is hardly ever the case. And that's why even in PTM proteomics, mass spectrometry is the best, most used method in the field, because with mass spectrometry, you can measure PTMs, because every PTM will induce a mass shift on the underlying peptide. So the masses will increase or decrease, and this delta mass, which is shown there for some PTMs in the table, is what we use to identify then this PTM. Okay, here you see the typical workflow that is used in bottom-up proteomics to study PTMs. And on the first slide, I can tell you this workflow looks very similar to what we call normal proteomics, where we measure proteins. And actually, this is also true. So this workflow is very similar. We usually always expect first the proteins, we enrich to certain proteins if we're interested in them, or we try to fractionate proteins to make the samples less complex. We then digest, we then enrich or fractionate peptides, we then do mass spectrometry and analyze the data. And this whole workflow for PTM proteomics is really very similar to what we always do. There are just a few exceptions, which I would like to highlight. And the first important point to consider when you want to do PTM proteomics is this protein extraction step, because there is one problem with not all, but some PTMs, which is that PTMs are transient and can change very rapidly. So we have reversible reactions ongoing. For example, for phosphorylation, we have kinases that attach the phosphate group, and we have phosphatases that remove them. Or in acetylation, we have the acetyltransferases and the acetylases. And these reactions can be very fast, within seconds. So if you use a protein extraction method that does not preserve the PTM status of your sample, which means that if you use an extraction method where phosphatases or kinases are still active and start changing the PTM status of your proteome, then what you measure in the end is very biased by your extraction method. This is not the case if you just look at protein concentrations, because protein concentrations just change so much slower. You need the whole ribosome machinery before you will see changes. With PTMs, this is different. So people try to work with extraction conditions that maintain this PTM status. And what people usually do here is they try to work with very strongly denaturating conditions by working with urea or clonidinium chloride or SDC, or with very strong acids, under which conditions these kinases and phosphatases, transacylases are all inactive, hopefully, and therefore do not change the status. Or people tend to work with inhibitors for kinases and phosphatases, which, if they're available, you can do too. Or they try to work very cool, under cold conditions, because this will slow down all reactions. Or they do the opposite, they work super hot to, again, denature every protein that is in there. Exactly, so the protein extraction method, which is really the first step of every workflow with PTMs, is really something very important to think about. Okay, then we have this PTM enrichment step, which is, of course, again, something that is very specific for PTMs. And this is also often a game-changing step in the project, because if you have a well-working PTM enrichment procedure, this can really drive a project. It can be very positive for a project, or can make your ability to detect these PTMs increase a lot. The prime examples of very well-working enrichment strategies that people use in the field are ubiquitin enrichments by working with anti-KJJ antibodies. These are antibodies that can recognize lysines that carry on their side chain a GG motif, which is a leftover from ubiquitin after digestion. So every lysine that was ubiquitin related will carry this GG on the side chain once you have used trypsin. There are also good antibodies available for acetylation of lysins, and there's also procedures for phosphopeptide enrichments using metal ions usually, and these metal ions will form a chelating complex with phosphate groups, and when you immobilize them, you can by that enrich phosphopeptides. So, and that's also the reason why in the literature these 3 biological PTMs are by far the best studied ones in the field of proteomics. Mainly also because we have such nice enrichment strategies. For a lot of other PTMs, there is no enrichment strategy available. So there's lots of room for young scientists to come up with great ideas and change this and make other enrichment strategies possible. Okay, then mass spectrometry. What is important to consider here for PTM workflows? Well, there's actually nothing really so different to consider. We use the same data acquisition types for PTM mass spectrometry as we would do it for normal proteomics. If we want to just identify PTMs, we use data-dependent acquisition methods. If we already have interesting PTMs or peptides, which we know, we use targeted proteomics to measure those very reproducible and sensitive. But you can also use data-independent acquisition or TMT-based quantification on phosphopeptides or on PTM peptides in general. However, what is, again, something to consider in PTM proteomics is that you can sometimes, depending on the PTM, exploit diagnostic ions that are formed by a PTM to either identify the PTM or to maybe also validate that PTM, because some PTMs might fragment during the fragmentation process in the mass spectrometer. So one example is phosphotyrosine. If you have a phosphotyrosine in your peptide, you will get such an ammonium ion here at the beginning of the spectrum very often. And if you see such a specific mass, such an ammonium ion, you can be very certain that there is a phosphorylated tyrosine in that peptide. Or when you have phosphorylation on serine or threonine residues, you often get this loss of this neutral loss of a phosphate group from certain fragment ions. And then you get all these fragment ions that show a mass difference of 98 Dalton. And then you know this was a phosphate loss. Okay, on my last slide, I want to just once more summarize what I think are currently the major challenges in PTM proteomics. And one major challenge that you have when you want to do PTM proteomics is site localization, which means if you want to say on which exact amino acid sits my PTM. For some PTMs, again, this might be very easy because maybe there's just one single amino acid in a peptide sequence that can carry this PTM, and it's more or less clear. But for other PTMs, this can be a real issue. The prime example here is often phosphorylation. Phosphorylation can occur on serine, threonine, tyrosine, and most peptides that exist in nature carry several serines, threonines, or tyrosines. So here's one example of the peptide sequence that can have 3 different serines, so 3 different potentially phosphorylated amino acids. And if you want to say in the end which of the 3 is the one present in your sample, you must use the information from the fragment ions that you see in this table, not very well, but in color code. You must use this information about the site determining fragment ions to differentiate between these different forms. And sometimes this can work well if the peptide fragments very well, but sometimes this can also be really tricky if these site determining ions are missing. Exactly. So another major challenge in PTM proteomics are missing values and limited reproducibility. So in the moment when we want to quantify proteins or identify proteins, we are usually in bottom-up proteomics in this luxury position that we detect very often several peptides of a given protein. And to identify and to quantify this protein, we can use the information from all of these peptides and use several data points, let's say, to identify and to quantify that. In PTM world, you work on peptide level. You always just have one single peptide that can tell you whether this PTM is present or not. So the data tends to be more sparse, with more missing values, random missing values, and issues with reproducibility. Also, sensitivity really matters for PTM peptides more than for normal proteomics. So PTM peptides tend to be of low concentration, very often also because of low PTM stoichiometry. So a a protein is not modified by a PTM to 100%, but maybe just 1% or even less, just 0.1%. So you have just a small concentration to aim for. And PTMs might also lead to low ionization efficiencies. So it might be that this PTM just hampers the way how peptides ionize. And this again leads to lower intensities in the mass spec, into more problems to detect these peptides. And one last major challenge, holy grail in PTM proteomics, is the differentiation of proteoforms. So if you're working on a project where you really want to say which different proteoforms of a protein exist in a sample, so how do different PTMs on the same protein maybe interact, communicate, crosstalk to each other, then you're not allowed to use a protease in your workflow and do what we call this bottom-up proteomics. Because in the moment when you use a protease, you cut the connection about which peptide was present in which proteoform. So there's no way in bottom-up proteomics for you to say something about the presence of different proteoforms. The only way to address this question is to measure the complete protein, the complete proteoform, amid working with proteases, and this is still very, very challenging to do. Okay, this is all I wanted to tell you as a starter about PTMs and mass spectrometry. Are there any urgent questions from the audience?